All right, so in the previous lecture, we talked about different factors that affect the enzyme activity. Number one was temperature and number two was pH. So now we're going to carry out a practical investigation in which we're going to check the effect of temperature on the enzyme activity. Now what we'll do is, first of all, we need a starch solution. ठीक है, हम एक starch, अब हम कर क्या रहे हैं? We are just checking the effect of temperature on enzymatic activity by carrying out an experiment. Number one, starch solution is heated to a set temperature. Iodine is added to wells of a spotting tile. Amylase is added to the starch solution and mixed well. Every minute, droplets of solution are added to a new well of iodine solution. अब क्या करेंगे? ये एक starch solution लिया इन्होंने and they have heated it. Then they have taken a spotting tile, एक white tile होती है, और उसके ऊपर उन्होंने क्या किया है? Iodine is added to the wells of spotting tile. उन्होंने iodine यहाँ पे रख दिया, ठीक है? 1 minute, 2 minute, 3 minute and 4 minute मार्क भी कर दिया, iodine यहाँ पे रख दिया. Now amylase is added to the starch solution and mixed well. अभी आपको यहाँ से heat भी दे रहे हैं starch solution को. Now starch solution is the substrate और आपके पास जो amylase है, amylase is the enzyme. तो आप temperature भी दे रहे हैं. And that enzyme is also being added. Now every minute droplets of solution are added to a new well of iodine. First minute में अब enzymic activity start हो चुकी है, temperature भी increase हो रहा है. What are you going to do? You take a syringe and a dropper and you're going to drop a part of the starch. आप क्या करेंगे? आप few quantity, आप थोड़ी सी quantity लेंगे of starch solution की, आप कुछ drops of solution के निकालेंगे हर मिनट के बाद and add it on the iodine जो आपने spotting tile. After 2 minutes, you'll again take some drops from the solution and you'll add it with iodine. Again after 3 minutes, आप यहाँ पे डालेंगे और 4 minutes के बाद you'll add it over here. This is continued until the iodine stops turning blue black. अब यहाँ पे भी blue black हो जाएगा, यहाँ पे भी blue black हो जाएगा, यहाँ पे भी blue black हो जाएगा. Let's say at 4th minute, the iodine drop does not turn blue black. इसका क्या मतलब हो जाएगा? इसका मतलब ये होगा कि now all the starch has been broken down. क्योंकि हमने एक food test पढ़ा था that for starch solution we use iodine solution. So when the solution is stopped turning blue black, this means that no more starch is present. All of the starch has been broken down by the amylase. तो आपके पास फिर वहां पे कोई blue black color नहीं आएगा. Time taken for the reaction to be completed is recorded and experiment is repeated at different temperatures. अब आप experiment को different temperatures पे करेंगे. क्यों? आप check करेंगे कि what does it have the effect on the time? क्या time पे फर्क पड़ता है कि how much time does it take for the starch to be completely broken down? Let's say at 10 degrees Celsius आपको पता लगा that it takes 6 minutes कि आपको 6 spot का क्या मतलब है कि 6 minutes के ऊपर there was no more starch present. Let's say जो आपने temperature increase किया तो 30 degrees Celsius, maybe at the third spot the blue black vanished. इसका क्या मतलब है that at a higher temperature less time was taken for the starch to be broken down. So the quicker the reaction is completed, the faster the enzyme is working. So a quickie, step number 1, we have a starch solution and we're going to heat it, for example, at 30 degrees Celsius. Then we're going to add a few drops of iodine on a spotting tile. जैसे कि ये 1 minute का हो गया, 2 minutes, 3 minutes, and at every minute I'm going to add. दूसरा मिनट गुजरेगा मैं उसका यहाँ पे ऐड करूंगा. जब 3rd minute पास होगा I'll add it over here. जब 4 minute होगा I'll add it over here. Let's say यहाँ पे blue black हो गया, यहाँ पे blue black हो गया, blue black, blue black, blue black stop. यहाँ पे orange ही रहेगी. This means कि 4 minutes के अंदर solution आपका totally breakdown हो चुका है. You're also adding amylase, starch solution, add amylase to the starch solution, start the timer straight away. Every minute, remove a drop of a starch solution and add it to iodine in spotting tile. Iodine turns blue-black, which means starch is present. Less color change, some starch is present. और यहाँ पे तो no starch is present. No starch is present in the last 8 minutes. Starch कब तक present थी? At the 3rd minute. 4th पे the reaction completed. So there was no change after the 5th minute, which means the reaction has completed. Then you repeat the steps 1 to 6 for different temperatures. अब यही वाला आपने different temperature पे करना है. Let's say at 20 degrees Celsius, it took 9 minutes for the starch to completely break down. When you increase the temperature from 20 to 40, it only took 2 minutes. And when you changed it further from 40 to 60, क्या हुआ? आपका reaction ही नहीं complete हुआ. It remained blue black. Why? Because the amylase died. The amylase was denatured due to very high temperature. Hence, starch broke down ही नहीं हुआ. Starch wasn't even broken down because आपके पास जो amylase enzyme थे, that must have denatured. At low temperature, amylase enzyme अच्छा काम कर रहा था. This was the optimum temperature because the enzyme worked the fastest. First minute में starch was present. At the second minute, starch was present in less quantity. At the third minute, starch wasn't even present, which means that all the starch was broken down by the amylase. यहाँ पे starch broke down ही नहीं हुआ है because amylase has been denatured at higher temperature.